Last week, we got an overview of how proteins are made, but this week we're going to get to dive into the nitty-gritty of this process called protein synthesis. Side note, to synthesize something means to make something. So protein synthesis just means making proteins. There are two steps of protein synthesis, transcription and translation. Transcription is going from DNA to RNA. And then the second step is translation. So translation then is going from RNA to protein. This idea that information goes from DNA to RNA to protein, it's called the central dogma in biology. It's a really core concept that information generally goes from DNA to RNA to protein. Transcription happens here in the nucleus. So we're going to talk about how transcription would happen in eukaryotic cells like ours. Transcription uses an enzyme called RNA polymerase. But RNA polymerase is the enzyme that will make our mRNA. So what it does is on DNA, there's these strands called promoters, and it's a specific sequence that tells RNA polymerase, hey, this is where you should start making a strand of RNA. So then RNA polymerase opens up the two strands of DNA, it separates them, and it starts to make a strand of RNA here. And how it does that is it uses the base pairing rules that we learned about. So you see here there's a T on the DNA. So this mRNA strand starts with an A. Then there's an A here, and we have a U because remember this is RNA. So it carries on making this strand of RNA until there's a sequence that tells it to stop making the RNA. The RNA strand that was made by RNA polymerase is not quite ready for step 2. It has to go through RNA editing. And in this step, portions of the RNA are cut out. So in going from this to this, you can see these red parts have been cut out and discarded. So those are called introns. The blue parts that are kept are called exons because they will be expressed as protein. So what we're left then after we cut out the introns is our ready strand of mRNA. It's ready to leave the nucleus and go on to step 2. An interesting side note, scientists don't totally know why we have introns or what their purpose is. It's something that research is going into to try and figure out why that is. So there's still a lot of things that we need to figure out about the world around us, which is one thing that makes biology so exciting. Okay, now this mRNA is ready to move from the nucleus out to a ribosome. Translation happens in the ribosome. So ribosomes can either be out here in the cytoplasm or attached to the endoplasmic reticulum. So translation happens in the ribosome. So this blue and yellow structure here is the ribosome. And so our mRNA strand, that's what this is representing, has traveled here from the nucleus. So it is at the ribosome now, our mRNA strand, and it can be read now by the ribosome to make a protein. So how does that happen? Well, the mRNA is read 3 bases at a time by the ribosome. So every 3 bases is what's called a codon. Now the first codon on mRNA is always AUG. That's called the start codon. And that tells the ribosome, here's where you should start making a protein. Okay? Now these should look familiar. These are tRNA molecules. So if you remember from last week, those tRNA, each of them is carrying an amino acid. Amino acids are the building blocks of protein. So when the ribosome reads a codon, then a tRNA that matches with it pairs up to it. So you can see these base pairs match. This says AUG. The tRNA has UAC. They match. And so this tRNA pairs and adds its amino acid. Then the ribosome would move to this next codon, UUC. This tRNA binds to it and adds its amino acid to the chain. Then this mRNA would keep moving through here. We would get to this next codon where this tRNA is going to bond and add its amino acid to the chain. And so the mRNA would keep moving through here this way, and the ribosome would keep adding amino acids to this chain until it reaches a stop codon, which tells it to stop making the protein. All right, so let's put it all together. So DNA to mRNA, remember that is transcription. So we were already practicing this last week, going from DNA to the mRNA strand that would pair with it. So what we need to remember is we need to remember the base pairing rules, that A pairs with T or U in the case of RNA, and C pairs with G. So then using those rules, we have an A here in DNA. Sorry, we have a T here in DNA. So it'll pair with an A. Then we have an A, so it'll pair with a U. Then a C, so we would have a G. Then a G, so we would have a C. Another C up here, so we would have a G. A T, so we have an A. An A, so we have a U. There's a G, so we put a C for its pair. Then there's two C's, so we'll have two G's. An A, so we have a U. A T, so we have an A. An A, so there's a U, and then two T's, so two A's will pair with those. Then going from mRNA to protein, remember, is translation. Now, to figure out what each codon, what amino acid each codon codes for, we use what's called a codon chart, which I have up here to the right. So let me show you how to do this. Remember, each codon is every 3 bases. So I'm actually just going to draw a line in here because that helps me. So our first codon is AUG. So in order to use the codon chart, you start in the middle. So this one's AUG, so I find A in the middle. Then I move out to the U. And then G, I move further out, and our first one is methionine. Now this is the start codon. Start codon is always methionine. So I put that, okay, that's the first one. Next, CGA, I'm going to draw another line to help me. That's my next codon. So I find C in the middle and I work my way out. C to G to A, that's arginine. So I would write that next. These are the names of amino acids in our body. All right, next one, UCG. So I come over here, U, out to C, out to G, that's serine. Then GUA, let's try it. Find G, U, A, valine. And then our last one is UAA. So we've got U here in the middle, we move outwards, A, and look at that. That is a stop codon. So it's no amino acid. It tells the ribosome, stop making a protein. So that is how we would do these 2 steps if we were writing it all out. Okay, so this is one more practice example, just to help you be more comfortable with this. So remember, we go DNA to RNA, then RNA to protein. This is what happens in our cells. So if I had this strand of DNA, first thing I would do is transcription, which is putting it into RNA that would pair with this. So T pairs with A pairs with T, but since this is RNA, remember it's U. C pairs with G. G pairs with C. A with U. G with C. A with U. A with U. C with G. Lots of U's because there's an A, then another A with U, then another A, and then another one, wow, and then a G, and then an A. So that now is our strand of mRNA. So now this mRNA will leave the nucleus and it will go to the ribosome for translation. So for translation, then we use this codon chart, okay? So let's start with this first one. I'm going to draw lines again to help us. AUG. We start in the middle here and we work our way out. So A, then you're looking for the U that's still in this A corner. So A, U, G, methionine. Remember, that's the start codon. Then next, we have C, U, C. So I find C, U, C, leucine. Then U, U, G. So I find U in the middle and I work my way out. U, G, another leucine. Then U, U, U. So U, 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 phenylalanine. It's a long one, so I'll have to write small. Phenylalanine. Okay, then last one, UGA. We find U, G, A, and that is a stop codon. So stop is our last one. Okay, so that's another example of how we go from a strand of DNA to a strand of RNA to now all these amino acids, which will make a protein.